maybe it's time to introduce Jürgen, which is the first talk of the day. So I'm contributes to computational tools and models that optimize metabolic pathways, emphasizing sustainable biotechnological application. His research aims to improve the efficiency of microbial production system using mathematical models to predict and refine bioprocess performance. And he works in impact metabolic engineering, synthetic biology, and green bioprocess development. So today he will present a talk on boosting biotechnological production process. So it's to you, Jürgen.
Well, thank you, Sabine. I hope you can hear me. Hope you hear me all right. Okay, excellent. You should also see my screen. And well, thank you. Thanks for having me. And yes, I'm gonna talk about boosting biotechnology production processes with constraint-based process modeling. What I'm going to do today is, well, first of all, I'll start with the acknowledgement because I typically run out of time. So the work I present here is essentially from three people, Matthias Gottsmith, Guido Schlögl, and Leopold Zertner, who worked on these tools that I'm gonna present to you. And of course, my group is a totally dry lab group, so we don't do any experiments. We have many collaborators that actually then try to validate whatever we predicted, especially some universities here in Vienna, the BOKU and the Technical University, but also some collaborators in Canada. And there are some guys that eventually paid the bill. Thank you for that. Okay, so what I'm gonna try to do, try to convince you today is that although we have like bioprocesses that are bioprocesses that are very well refined and have been, especially in the biopharmaceutical industry, for instance, trained and refined and optimized. If we're going to use rational models or rational tools that allow us to even further optimize these processes, there is a lot to gain. And what I'm trying to convince you that even if you have a process like this one that is far or much better than everything you find in the literature, if you use rational methods, there is still a lot of that you can gain. And I'm gonna try to convince you that this is an example that I'm gonna show you in a couple of minutes. That's a plasmid DNA process that we optimize. And I'll try to focus here that this really is a process that has been going through a lot of experimental validation and refinement. But if we're gonna do some more computation, there's still a lot to gain. In order to do this computation, I'm gonna introduce two software tools that we have developed in the recent years and that just came out this month. One is a dynamic control FBA analysis tool that allows you to design a bioprocess, and also a tool that's called OptFed that allows you to design a fed batch process in a realistic manner. I'm going to show you some applications for butadiol production, plasmid DNA production, and protein L production. And I hope at the end of this talk, you're going to say, well, yes, that's something we should do and we should keep developing. Okay, but let me start with this first example, butadiol. What is it? Well, it's a small molecule, and it's essentially a precursor for a lot of different different products in food applications, as a jet fuel, in cosmetic and biopharmaceutical. The key thing here is, of course, this molecule is nowadays produced by petrochemical applications or petrochemical way of producing it. And if we want to be able to turn around or to turn our processes into a sustainable form, we have to beat this number. So that small molecule has a price tag at the moment of of roughly $4 per kilogram. So that's really a little. Our process, if we want to have a sustainable bioprocess, needs to be really, really efficient in order to beat this number. How efficient? And is it even possible? There have been some studies, some techno-economical studies, that this number can be beaten. And there are a couple of constraints or a couple of conditions that have to be met. Well, first of all, we have to utilize cheap raw materials. The cheaper, the better. And ideally, those cheap raw materials come from inexpensive waste stream. We'll need to develop an optimized production host that is able to obviously utilize this cheap raw material and produce the product of interest with a high yield and a high efficiency. So in terms of these cheap raw materials, there have been a couple of papers and studies that try to address this, also in terms of what kind of organism we're going to use. And these 2 points roughly get us half the way. The other half comes from the bioprocess. So we need to implement efficient bioprocesses that maximize productivity and minimize cost. And this is where we can gain half of our price tag. And in order to do this, there are some studies that have actually calculated some key numbers that have to be met. And in terms of our bioprocess, that we need to achieve a titer of 50 grams per liter and a productivity of at least 2 grams per liter an hour. Is this possible? And then finally, of course, if we want to be environmentally sustainable, our process should also reduce the emission, and we need to take into account this whole life cycle of the process. What I'm going to focus today is exactly at this third point, implementing an efficient bioprocess. Okay, so what's the current situation? So here is an example of the butanediol production process. Essentially, you have a bioreactor and it's run in a fed batch fermentation. That means you first inoculate your bioreactor, then you feed it with some substrate. In our case, we are only interested now on the bioprocess, so not on the raw material. So we use glucose, we use E. coli, but in principle, we can use anything else if available. So then we feed the cells, put in glucose, and we increase the cell density. And then if all this initial glucose is consumed, we start feeding the cells and further produce the product. And essentially, we have two parameters that we can vary. We can design the feed, so the supply of fresh glucose or fresh substrate, and we can modify the oxygen rate that influences the butanediol production. And of course, the cells also respond to this feed medium and to the oxygen restriction. So we need to model the metabolism, which we typically do by flux balance analysis, and we take into account the feed and the oxygen. So what's the process that, or what's the properties that this process has? Essentially, it strongly depends on the oxygen supply. Under aerobic conditions, we have high growth, but very little production. We have already an optimized strain that has some mutation that increases the yield of the butanediol. And this strain is not able to grow anaerobically, but what you can do, you can grow microaerobically. And then the growth is reduced and production is up. So here is the experiment, how a typical process looks like. It's essentially a two-stage process. We first increase the biomass, and at a certain point, you switch to microaerobic condition, and then you start producing your product of interest. Okay, the question is now, is this the best possible process? And what we're going to do, well, here is our matrix. We have on the x-axis the titer and the productivity. I told you these dashed lines, those are the boundaries that design or that bound our economically feasible process. So we need to be larger than 50 grams per liter, and we need to have a productivity of larger than 2 gram per liter an hour. So this is the area, the target area where we need to have our process. The process that is depicted here is this point. So we do have a high titer, but our productivity is just by a factor of two too little. Can we improve this? And if so, how can we set the control variables? How can we influence, or how can we determine how the feed, how the oxygen has to be as a function of time to reach this economically feasible process? If you are in the constraint, if you are from the constraint-based field, you know you can do such process simulations, and the key to it is our dynamic FB flux balance analysis simulations, and they are well established. What does that mean? Well, essentially what we have is we model our cell in terms of a linear program. That is, we optimize metabolism, typically some growth rate, subject to steady state conditions. So all these here are the intracellular fluxes, and these intracellular fluxes have some bounds. They have either bounds as we take up nutrients from the medium, so they are influenced, or these bounds are influenced by the control variables that design or that make up our bioprocess. The control variables in this case is the feed medium and the feed flow, and also the oxygen supply. And okay, then we just integrate these exchange fluxes, the nutrient uptake and the nutrient secretion, or the waste product secretion in the reactor, and integrate this up, and that's basically dynamic flux balance analysis. That's a method that's quite old. How can we then use this in order to optimize this? Because what we want to optimize, well, of course, we have to optimize the metabolism, but actually what we're really interested is we are trying to optimize the process. So basically, we put on that FBA solution, or on this FBA problem, on top of it, we put another optimization principle, and whatever we want to optimize. We could optimize for productivity, yield, titer, or yield, and we optimize this by determining our control variables, so the feed and the oxygen supply. Okay, now you know this is now becoming a nested problem, and that's difficult to solve. There is some progress in turning a nested optimization problem into a single level optimization problem, and the way to do this is via the KKT, the Karush-Tucker constraints that allow us to turn this nested optimization principle into a single level optimization, but we make it, first of all, much larger and nonlinear. So this is impossible to solve, but this now is being solvable. But it takes a lot of resources, especially if you think we have a genome scale metabolic model that has, like, say, 3,000 reactions. And if you want to solve this, we get, for every constraint that we have in our model, we get an additional variable. So this model, this system that we actually have to solve is really huge. So what we did is we looked at how can we accelerate solving this problem. And one of the very obvious things is very simple. There are a couple of tricks Matthias did in his tool, but one is very easy. That's why I'm showing it here. So basically, the question is, we have many constraints, thousands of constraints, but are all of these constraints limiting? Especially if you think on a standard genome scale metabolic model. If you go to the big database and download any model, what you get is you get bounds on every reaction. And these bounds are kind of the diffusion limit. And here's a simulation. Actually, these diffusion limits, these default bounds in these SPML models or in these COBRA models, is never actually constraining or never limiting. But the solution space is much more, so even more exaggerated. So basically, we can just simply pre-process the constraints and screen for bounds that are not constrained. If we do this, just simply by removing the default bounds, 10,000 and minus 1,000, we already reduce the simulation by half of the, so half of the variables in our simulation are then no longer needed. And what we get is we get a reduction in runtime of 80%. So basically, this is our starting point, and then we cut it down the runtime to 80%. And that helps us a lot because now we can run these simulations in essentially no time and repeat that over and over and over again. This is what we do in the following. So basically, we now have not only a dynamic FBA, but a dynamically controlled FBA. And this allows us to identify the optimal solution space. Essentially, we can identify all solutions possible, but of course, we are interested in the optimal performance. So here is our starting point. Here is this process that we started on. We have a high yield, but low productivity. And now what we can calculate is we can calculate all possible processes. And you see here, this blue area are all the biologically feasible solutions, and the black line are the optimal solutions. Okay, that looks good. And it already looks we can get very close to the economically feasible solution. How does a simulation look like? So here is a high yield solution. And basically, this is a solution that has lower yield, but higher productivity. And what we find is, okay, we need to do two things. We need to make the process time or the process length shorter. And also we need to increase the biomass. So we need to increase the ratio between the first stage and the second stage. Okay, good. So let us look more into these solutions. So here is the solution space from before. And what we find is all the optimal solutions. So let me point out that our simulation does not suggest or does not require that the optimal process needs to be a two-stage process. But what we find is it always produces the best process if it has two-stage. Two-stage means first we grow biomass and then we produce the product. Okay, there are two areas. One area is over here where the process time is short and both productivity and titer increase. Here we have the maximum. So at a process time of 22 hours, we achieve the maximum productivity. And this maximum productivity is achieved at the lowest possible growth rate. And finally, if we are over here, we are in a trade-off region. When we increase the titer, productivity decreases. And you see this here as well. So here are the processes that are too short. And over here, that's the productivity. If you increase the process length, the productivity decreases, but titer increases. Okay, good. Yes. Now we can not only calculate the feed profile, we can calculate when we need to switch from stage 1 to stage 2, and we can also calculate the optimal growth rate that we adjust with our feed. And of course, there was someone from our partners that did the experiment, and it worked out really, really nicely. So here again is this curve that I showed you multiple times. We started here, and the optimal bioprocess that we designed really comes now extremely close to what we should have in an optimal bioprocess that is also economically usable and sustainable. So basically, with this dynamic control FBA, we can accurately design a bioprocess, a multi-stage bioprocess. Okay, but does it also work for other processes? And one other process is plasmid DNA that we looked at. Plasmid DNA is some piece of circular DNA that is produced here, for instance, in E. coli. And of course, you know it from the COVID vaccine, but also for gene therapeutic and cell therapeutic application. This element is going to be in huge demand in the coming years. So whatever we can do here in increasing the bioprocess is very useful. Again, we want to design a production process for this plasmid DNA, and it could be a multi-stage production process, but we don't really say that in the beginning. But essentially, what we would have in a multi-stage process, we have first a high growth to high biomass, and then we switch where we don't grow anymore, but use a production stage. If this is possible for plasmid DNA, there are two things that we need to find. We first of all need to find the decoupling agent that tells us when to switch or how to switch from here to here. And we need to determine when is the best possible option to switch. Okay, for this, we did FBA and essentially looked at what nutrient requirements do we have for individual functions? What do I mean by the individual functions? So these black bars are the nutrient requirements for biomass production. And you see, okay, that's minimal medium, and you essentially need all of this minimal medium. Good. For plasmid DNA, you don't need all of them. You essentially need these guys over here, the red guys. So basically what we could do is we could limit on any of these black bar onlys, and that would introduce a switch from the growth stage to the production stage. We looked into the literature, which of these nutrients is useful, and we figured, well, sulfate is a good candidate because literature says that's a metabolite that doesn't interfere with energy metabolism. And so the hope is, okay, if we limit sulfate, we can stop or we can achieve growth arrest and switch on production. Why would we think so? Well, here would be the idea. We have some productivity or some plasmid synthesis flux, and now we have the sulfate limitation. So here is the sulfate depleted. And basically what we think, what happens, well, all the fluxes, all the enzymes are expressed. There is some limitation in the biomass growth. What should the cell do? Well, it takes the carbon that is there and pumps it now into plasmid because this is already expressed. And so basically we have now a parameter that we somehow represents the synthesis boost upon limitation. And essentially what I'm going to show is if this kappa is larger than 1, we get a two-stage process. And what we find in the simulations is as soon as this kappa is positive or larger than 1, then we always get an improvement. So this is what you see here for different values of kappa. So for different values of the synthesis boost, we get different optima, and we have to starve it at different. So on the x-axis, you see the length of starvation that we have to starve this process, and then we get different optima with increasing kappa. And here is a time simulation. So basically you see, okay, here is the growth of the biomass, then the sulfate is depleted and the biomass goes down because we keep feeding, we keep adding volume, so it's diluted out. But you also see that the product is further produced, and we see an increase in the specific plasmid yield. Okay, that's our idea. We went into experiment and exactly what we saw. This is what we saw. We do see that this principle works. Here is the dilution, here is the increase, here is the standard process. But the very important part here is the plasmid by biomass, and we get a yield, an increase by 30%. Well, that's very encouraging. We went back into our model and checked, okay, how is the synthesis flux? Remember, we assumed that it's a constant and then jumps. We don't see that in the experiment. We see a decline, a continuous decline, but we do see this jump, and that's important. And so that's good. We don't really know at the moment why the synthesis rate is constantly increasing, and this is still something we want to investigate in the future. Especially we want to identify how can we prevent. We want to keep it as constant as possible as we now model. We look further then, if we do this processing and look at the experimental data, not only do we find that the plasmid yield increases, but we also find that the quality increases. The reason for this is not really clear to us, and still something that we investigate, but we see here is the starvation, we get a higher quality product. So not only do we get more, we also get a better product. Okay, now here's the process I showed you in the beginning. Here are the data points. Here are some batch simulations that we find in the literature. They are not in any way compatible and not in any way economically feasible. We have fed batch processes, and up here is the process where we started. And even for this process now, we can introduce this gross decoupled fed batch procedure that allows us to increase the specific yield by 30%, and the average volumetric productivity is still by 30%. So as I have been pointed out in the beginning, this is a process that is highly optimized, but we can still do significantly better. Okay, now, of course, does this limitation, this sulfate limitation work for other products as well? We looked at different possible metabolites that could be produced in such a way, and here is essentially the KEC pathways, and all the red dots mark targets for metabolites that could be produced in a sulfate limitation manner and should benefit from this intervention. Okay, last example, protein L production. So I've shown you that our method works on small molecules. We have shown it works on a plasmid, which is some macromolecule, and now we look at proteins. What was the problem? Well, essentially, our problem so far was we had this wish. So we had the model assumption was that the plasmid synthesis flux is constant and then jumps. Okay, in reality, we saw, well, that's not constant. And so basically we had to go back to the model, change the model, and blah, blah, blah. So the question for us was, can we actually integrate all the data that we have already in the design process before we even start modeling? So basically, what we tried to do is, and that's Guido's work, he developed a tool that's called OptFed that allows you to design a bioprocess by using some mechanistic insight that you already have in the process, but is essentially based on the data you have. And basically what we do is we have a very general model, and we train or we fit this model to the data that is available. We identify all the necessary and key information of these models and disregard all other parameters. So we basically reduce the model, and then we optimize and come up with an optimized feed profile or an optimized bioprocess. So basically here is the example that we did. We had experimental partners that did a response surface methodology to design a bioprocess. So these are all the experiments they had. So the star formation, and this was the input for our OptFed algorithm. And basically what we got out is, okay, we got out something that looks like this, rather than having the optimum as in the response surface methodology close to this middle star point. We found, well, actually this whole bioprocess has its optimum far away, somewhere at the edge of this process or of this design space. Well, is that true? So we went into the experiment, and this is what we got. Basically, you need to compare this red line with the black line. So we designed a bioprocess, and our aim was to, again, optimize the specific yield, so this is the product to biomass yield, and this increase here represents an increase by 90%. We looked into our model. So basically our model said, okay, run this process at 36 degrees, use a almost linear feed, and you should get a 21% improvement. In reality, when we did this simulation, when we did the experiments, we found that it's pretty close, so we got 90% improvement. So you see, so this is up to the point here, and then we had some problem in the infrastructure of the experiments. Basically, we were not able to cool the reactor any further, and then the results went off the bit. Okay, so now that brings me already to the conclusion, and I hope I could convince you and I could show you that the rational design of bioprocess has still huge potential even in a highly optimized process. I introduced two tools that we recently developed, and they are published in October. It's a dynamic control FBA and OptFed. Both help you depending on either a more mechanistic approach or on a more data-driven approach to optimize your bioprocess. Try to convince you, and I gave you some examples, one for butadiene production, where we increased the productivity by 104%, one for the plasmid DNA, where we had the specific product yield up by 30%, and for protein L, where we had the specific protein yield up by 19%. Here are the figures. Remember, for plasmid, we use the sulfate, which could be a limitation, which could be very useful in other applications as well. Here is the example for the butadiene, where we almost get into this feasible space. And finally, here are the results where we talk about the protein L. Well then, that's it from my side. Thanks a lot. There's people, funding agencies I want to acknowledge, and please do check us out. Here's our website, and I'll be looking forward to any feedback you have or any other question. There's one or two more announcements that I want to make. We were lucky to got funded quite recently, so there's going to be a cluster of excellence for cellular bioengineering. And in the next couple of months, we're going to post 20 postdoc positions and 50 PhD positions. So if you are interested in working on cellular bioenergy, please check out the website. It's circularbioengineering.at, especially if you're interested on this modeling part. In my group, there will be a postdoc and 2 PhD positions. They're going to be free, or they're going to be posted in early January. So if you're interested, please get in touch now. You find all the contact details on my website. And finally, in July of next year, we're going to have the Metabolic Pathway Analysis Conference in Vienna. And I hope I'll see you there. And I'm looking forward to your questions. Thanks.